We'd like to welcome you to the World Food Prize Laureate Lecture Series. Usually this is an event that takes place during the World Food Prize Week events here on campus, but just along with everything else, COVID has completely turned things upside down and we're doing things a little bit differently. So this year, the Laureate Lecture Series is actually taking place outside the traditional week of World Food Prize events, as those events moved virtual as well. And this gives us some time to spread these events out so we weren't overwhelmed with virtual events throughout. We're glad to have you here. In a few moments, we're going to be hearing from Dr. Matthew Rouse and the great work that he has done and continues to do. And with that, the chat box is going to be open for you to drop questions in. So make sure to have that ready to go. We know some classes are joining at different campuses. We know some of you are getting together at apartments with roommates. If you're in one of those settings, it might not be a bad idea to designate a chat champion, someone who's going to drop those questions in the chat for the group. But we'll just keep that open. And with that, I want to welcome Dr. Matthew Rouse, a plant pathologist who is at the USDA Agricultural Research Services Cereal Disease Laboratory and serves as adjunct associate professor in the Department of Plant Pathology at the University of Minnesota. As part of the Cereal Disease Laboratory, Dr. Rouse's research mission is to reduce losses in wheat and barley to major diseases, including stem rust and leaf rust. His excellent team members at the CDL and University of Minnesota and international collaborators worked with Dr. Rouse to make a rapid progress in identifying new stem rust resistant genes, establishing new field disease screening nurseries in Ethiopia, and assisting breeders in release of stem rust resistant wheat varieties across the globe. In 2018, Dr. Rouse received the World Food Prize Dr. Norman E. Borlaug Award for Field Research and Application. And I believe just recently, he'll correct me if I'm wrong, the beginning of this month with a great team, he was awarded the Borlaug Gene Stewardship Award as well. So with that, we'll welcome Matt to the stage, and he should be able to turn on his camera and audio. And Dr. Rouse, it's all yours.
All right. Thank you very much, Brad. Really appreciate this opportunity. It's an honor for me to be able to speak with many of you. And I'd like to thank the World Food Prize for setting up this lecture. And I see Ellen Franzenberg online. So thank you, Ellen, for helping out with that. And then I also I see Tessa Mahmoudi online. And so Tessa worked with us some time ago. And so thanks for joining us, Tessa. I'm going to share my screen here. And talk about some of the research that we've done at the Cereal Disease Lab. And through this presentation, there are two themes that I'd like to emphasize. The first one is technology and the use of technology. And so, this is kind of a case study for the use of technology and how we can think about using technology going forward, especially considering genetically modified things and gene editing, and those things are certainly questions to consider for the future. And the second theme I'd like to highlight now and for you to think about through the lecture is international collaboration and teamwork. And so be thinking about maybe how collaboration and teamwork contributed to some of these things and how you can do that in your own setting. I'd like to tell you a little bit more about myself and where I work. I work for the USDA, the Department of Agriculture, Agricultural Research Service. And so in this map, you can see 90 locations across the United States where USDA ARS scientists work. And I'm located at the USDA lab in St. Paul, Minnesota, on the campus of the University of Minnesota. And across the US, there's about 2,000 scientists that work for ARS. This is a picture of our building, and we, as Brad said, we work on preventing yield losses to wheat, oat, and barley to diseases that threaten the US wheat crop. And in this presentation, I'm going to briefly introduce wheat stem rust, and it's really impossible for me to not talk about Borlaug when I introduce my research because of Borlaug's contribution to the field. After that, I'm going to mostly talk about genetic resistance to the disease stem rust that affects wheat. And so I've divided that section into two parts. First, the last 10 years, and then the second part, kind of the future and some recent progress that we've made, and then end with some conclusions. So first, an introduction. And I'd like to start by showing this map of where wheat is grown in the United States. You can see it's grown all over the United States, not so much in Iowa. You guys are pretty specialized there in a couple of crops, but a lot of other places, and really in the northern Great Plains, there's a big wheat growing area. The southern Great Plains and also in the Pacific Northwest are kind of the three main growing areas, but there's also wheat all across the United States. Now that wheat in the US corresponds to different market classes. And so the wheat that's grown in the different areas is used for different things. And so in the north, there's wheat in red here that's used for making pasta. And also the wheat in blue is used for mixing with other lower quality wheats for making bread. And then if you go to the southeast, most of the wheat is used for cookies and cakes. It's kind of lower quality wheat. And these different market classes correspond to different types of wheat that are grown in wheat fields. And there are even some of them different species of wheat. And so wheat is quite a complex crop in the United States, different species, even different market classes, and it's used for different things. This chart shows the impact of a stem rust epidemic on wheat from 1935. And so what the numbers in these states represent is across the entire state, the percentage of the wheat production in that state that was lost to stem rust. And so in North Dakota and Minnesota, you see over 50% of the wheat was lost to this single disease. And this is significant because basically it's knocking out or wiping out a couple of important market classes of wheat, the hard red spring wheat and the durum wheat. And so this disease, when it gets going, can be quite devastating. And if you look in this chart, it shows kind of the history of when these stem rust epidemics have occurred. So in the x-axis, you can see the year starting from around 1918 going up through to the present. And then on the y-axis, the percent of the total US wheat crop that was lost. So 1935, nearly a quarter of our entire wheat crop was lost to stem rust. Some of the other epidemic years, between 5 and 10% of the wheat crop was lost. And so, and another thing is that there hasn't really been a big epidemic for decades. Now these epidemics were caused by different strains or races of the stem rust pathogen. Stem rust is caused by a fungus. And so in 1935, there was a race 56 that we call MCCFC that caused the epidemic, and then a newer strain caused the epidemic in the 50s. And so what happens in the field is that wheat breeders release varieties that have different stem rust resistance genes. So like SR5 is a stem rust resistance gene. Those genes remain effective for some period of time, but the stem rust pathogen adapts to those genes, rendering them ineffective, and then you get a stem rust epidemic. And then breeders release new varieties with new stem rust resistance genes, and those remain effective for some time, but then you get a new race, and that causes an epidemic. And so in the United States, the genes that we have been using have been effective for several decades. At this point, I'd like to start talking about Norman Borlaug. And so you probably are very familiar with him, and I'm going to kind of talk briefly about his biography in the context of the Green Revolution and wheat and even stem rust. So Borlaug was born in 1914. He went to the University of Minnesota and earned a bachelor's degree in forestry in 1935, completed his PhD at the University of Minnesota in plant pathology in 1942, and then went on to join the Mexican program of the Rockefeller Foundation at the organization that became CIMMYT, or part of the CGIAR, International Agricultural Research Group. So there he was employed as a wheat breeder, developing wheat, and he made some progress, but really he obtained this Norin 10 dwarf wheat, a really short wheat line. And that was very important for his wheat breeding, because it takes about 10 years to breed a wheat variety. So some of those wheat varieties that he developed were distributed several years later across the world through a USDA nursery. And they were grown in India, a few locations, and then based off of how promising those wheat lines that Borlaug developed were, 450 tons of wheat were imported to India and Pakistan around 1965. And this isn't wheat for human consumption. This is wheat for planting in fields to grow new varieties. And so Borlaug's wheat that was disease resistant, shorter, so it could be fertilized more and irrigated more, was spread throughout South Asia. There were 18,000 tons imported into India of a single variety, Lerma Rojo 64. And again, thousands of more tons imported into Pakistan and Turkey in the late 1960s. This resulted in Pakistan becoming self-sufficient in food production in 1968. And this is significant because in that same year, Paul Ehrlich published a book called The Population Bomb. And it was well known and publicized that if something wasn't done, there was going to be mass starvation across South Asia. So because of these new varieties and agronomic practices, Pakistan became self-sufficient. Norman Borlaug was awarded the Nobel Peace Prize in 1970 for averting this disaster. And then a few years later, India became self-sufficient. So in a nutshell, that's really the Green Revolution. And you can see on the chart, there wasn't much going on with wheat and stem rust, but in 2005, UG99 arrived on the scene. And what Borlaug saw when he went to Kenya and looked at some wheat fields in 2005 were some pictures like these. And there were wheat fields destroyed by stem rust. You can see the wheat stems here, severely infected with stem rust, lodging of the wheat fields. And this wheat crop here, you can see wheat spikes, but inside those spikes are very small shriveled seeds because all of the nutrients have gone into the rust spores and not into the seeds. So it's basically wiping out the wheat crop in Kenya. Those seeds are so small that the people that use combines basically didn't harvest anything because the shriveled seeds just blew out the end of the combines. So because of this, and because Borlaug witnessed a lot of the epidemics when he was in Minnesota and Iowa in his growing up and in his college years, he kind of wrote an article in the New York Times warning the world that this is a serious threat to wheat production. It hasn't been a problem for decades, but if this disease spreads, it could become a big food security problem to wheat because this new strain is virulent to the genes that have been effective for a long time. So Borlaug penned this article in 2008, and then he passed away in 2009. So Borlaug kind of started with wheat and worrying about stem rust and ended, calling the world to pay attention to this disease again. And so what is UG99 and why are we working on it? And so again, this was highlighted by Borlaug as a threat. It was first identified in Uganda in 1998 and isolated in 99, hence the name UG99. first caused an epidemic really in Kenya 2004 and 2005. And it's a race group or a strain group of the stem rust pathogen Puccinia graminis. And why we're worried about it is kind of showed in this chart here. So there's a number of wheat lines listed here. They each have a different stem rust resistance gene. This picture shows the reaction of an isolate of the stem rust pathogen on these different genes. So just glancing at the pictures, you can see that there's a lot of infection and most of these genes are not effective. And so these scores kind of represent whether the lines are resistant or susceptible. So you can see the gene number 36 is resistant to this isolate of UG99. And we know this, these genes and the response on these genes is how we get that letter code. And so when you have the first four set of genes becoming susceptible, that corresponds to what we call a high reaction or high on all four genes. That codes for a T, hence the name TTKS across all of the genes, with just a couple of the genes maintaining some resistance. So there is more problems with UG99 because it keeps changing. And so as it has spread, it has also become more virulent. And so that gene 36 has become susceptible as UG99 has adapted to wheat varieties that are grown. Other gene, SR24, is now ineffective as well. So it keeps changing, becoming more virulent and spreading. And that spread is shown here. So in 2005, it was mostly just in East Africa. There was a related strain in South Africa. By 2008, it had become more virulent, a lot of newer strains, and spread into Iran. By 2014, additional strains, more virulence, and now it really hasn't spread much, which I'll talk about why it hasn't spread as much, but there are about 14 different variants of the pathogen right now. So we kind of know about UG99. It's caused by this fungus, and it's also a threat to wheat production. And I'd like to emphasize that in the context of the United States. And so going back to our US map, you can see here, again, the green areas where wheat is mostly grown. And the wheat germplasm in those areas is shown in pie charts. And so the red proportion of the wheat lines is the wheat lines susceptible to the original UG99. And then some of the variants of UG99, TTKST, TTTSK, providing virulence to additional proportions of the germplasm. So there's a small sliver of green in many of the important wheat growing regions in the United States. What this means is that if this pathogen that's in Africa and the Middle East would come to the United States, we could be in a situation like the 1930s or 50s where we have a susceptible crop, a virulent pathogen, and we could have some devastating epidemics in the right year. And so this is why we're working on it and at the USDA, and why people around the world are working on it together. So that was the introduction, stem rust, Borlaug, UG99. And now I'm going to talk about what we're doing to try and develop genetic resistance. And what this section is going to emphasize is two different things. First, genetic resources, so resistance genes. And then the second part, 3 case studies from around the world. This chart shows the 21 chromosomes of wheat. And around 2007, when I started working on UG99 genetic resistance, there were about 20 stem rust resistance genes that we knew were effective to UG99. So those genes are shown on different chromosomes in this chart. Now, some of the genes are black and some are red. The red ones are from different species than wheat. They've been crossed in from wheat wheat relatives. And so a lot of times that means there is linkage drag associated with them. So there's a big red X next to some of them. So basically they can't be used because they're on a big piece of a chromosome from a different species of wheat. And only a few of the genes have this blue M next to them. So we didn't have very many molecular markers that we could use in wheat breeding to help facilitate utilization of these genes. So since this time, we've more than doubled the amount of genes that are effective to UG99. So we've done a ton of genetic research. You'll see that the number of blue M's has increased dramatically. So we have molecular markers linked to many of these genes. There are green circles now. So several of the genes have been cloned. So we know the DNA sequence of those genes now. And the red X's have disappeared. So we've reduced the unwanted chromosomes regions around many of those genes. So we have a ton of genetic resources that we can use now. The red circles just show how my lab at the USDA has contributed to a lot of that progress. Not only do we have a lot of genes, we also have a lot of UG99 resistant varieties that have been deployed. And so this shows the resistant varieties that were released and spread around the year 2012. And so by that time, the wheat that was grown in the world in the immediate path of UG99 was becoming resistant. And I think that contributed substantially to slowing the spread of UG99 because we had resistant varieties in the field. But there were some problems. And so this picture shows a field of wheat in Ethiopia in 2013. And you can see there's kind of two sides to the field. One side was sprayed with a fungicide and one side was not. The side that was not sprayed was killed by stem rust. And this variety, Bigelow, was the most common variety grown in Ethiopia at the time, accounting for about 50% of the wheat acres in Ethiopia. And so there was this epidemic raging in 2013, and we wanted to know, okay, so did UG99 change again and become virulent to this previously resistant variety, or what happened? When we got samples of the stem rust pathogen in our lab, what we found is that there was a completely new race unrelated to UG99 that caused that epidemic. And this new race was virulent to a gene called SRTMP that was maintaining resistance to UG99, but the new race was able to overcome that resistance. And so this kind of woke us up that we can't just worry about UG99, we have to monitor and prepare for the other strains of the pathogen that were in these countries. Similarly, unfortunately, in Kenya, there was an epidemic on the most popular variety, Robin, in 2014. And so that variety was grown over about 67% of the wheat acres in Kenya. It became susceptible. This time it was a race that was in the UG99 race group. So UG99 had adapted and become virulent to that SRTMP gene again. So we have a lot of resistant varieties that were out in the field starting in 2010 and beyond, but a lot of times those resistant varieties just had a single stem rust resistance gene. And so we saw some of those varieties go down as UG99 was adapting and becoming more virulent. That latest strain of UG99 is particularly dangerous for the United States. And so I'd really like to highlight this group of germplasm here, the US spring wheat breeding lines. So out of the lines that we tested in 2014, about a third of them were resistant to the UG99 variants at the time. But when we looked at the new strain from Kenya, TTKTT, we went back down to a very small proportion of resistant varieties in the US spring wheat. And this is kind of bringing us back to the beginning. And so we made some progress in getting resistance in the United States starting around 2014, but the new race brought us back to where we were in 2005, 2007. So it just highlights how dangerous it is when the pathogens change. And then the final example here is in Italy in 2016, there was a completely different strain of the stem rust pathogen that caused an epidemic in Sicily, Italy. And this strain was virulent to the gene SR13B, which is important in pasta wheat or durum wheat. And this was the first time that stem rust had caused a problem in Europe for about 50 years. So we see that stem rust is on the move. UG99 is becoming more virulent. And we have a lot of resistant varieties and resistance genes, but those genes aren't making, a lot of those genes aren't making it into the varieties. So that was kind of a highlight of the past 10 years, unfortunately, where things were going. And now I'd like to talk about our recent progress and then going forward, what we're doing and maybe what we could think about doing. And this section is really divided into two parts: appropriate phenotyping or appropriate testing of wheat lines, and then also linkage blocks. And the key behind linkage blocks is using more than one gene. We know that when we have just that one gene in a wheat variety, it's very easy for the pathogen to adapt to it. And so similar to like using a single antimicrobial resistance, we know the pathogens can adapt to that quite easily. So for appropriate phenotyping, I'd like to show this picture of our screening facility in Kenya that we work with the Kenyan government, and then also Zena Koski, who worked for the Kenyan government and then came to the University of Minnesota for her PhD with us, and now is back working for the Kenyan government again. But this is really the main place in the world where screening for UG99 in the field is done. We do some work at our biocontainment lab here in Minnesota, but you really have to go to the field to see what's going on. And so because of this screening facility, we have been able to make some progress. And that is highlighted here in this picture of a wheat field in Minnesota. And so using the Kenya testing, we were able to release a variety of wheat called Linkert through the University of Minnesota. And this variety is resistant in the field in Kenya, but it's susceptible as a seedling. And what that means is that it doesn't have a single resistance gene. It has a different type of resistance called quantitative resistance. And so this is resistance that we believe to be and has been shown to be more durable and won't break down. And so this variety has been quite popular in Minnesota and North Dakota. It's been grown on nearly 1 million acres per year recently. And so we've kind of changed the game in the past couple of years, where now we have some resistance in the field, in farmers' fields, that's effective to UG99. So we're protected at least a little bit if UG99 were to come to the United States now. But if you think about all of the changes in the stem rust pathogen and what we saw in Ethiopia, where it was a completely different strain, we realized that we had to work with these other strains and not just UG99. This picture shows a couple of, or 3 people who worked for the Ethiopian government. Orku and Ndale came to the University of Minnesota to earn their PhDs. I saw Ndale in the lab here this morning, and they're standing next to the Borlaug statue at the University of Minnesota campus. And so we really started working more with the Ethiopian government after that epidemic in Ethiopia. And what we did is we isolated several different virulent strains of the pathogen and increased them, and then established a screening facility where we had different single race nurseries, where it was a small nursery with wheat lines inoculated with a specific race. So that allows us to look at what lines are resistant in the field to these different strains and not just UG99. And so here's a few pictures of the first nurseries that we set up. in Ethiopia. And what these nurseries allowed us to do was to look at the response across several different races. And so this chart shows data from the first couple of years. There are a large number of wheat lines listed here, different breeding lines, some varieties, and then their response to stem rest to these different races is on this axis. So really resistant reactions are over here, and really susceptible responses are over here. And these are actually the 50 most resistant lines in the nursery. And so what you see from this chart is that, a line might be kind of resistant to one race, but quite susceptible to another race. But we do see, around 20 or so lines that were resistant to the 4 races. Unfortunately, almost all of those 20 lines had a single stem rest resistance gene, SR24. And so we know that it would be unwise to use those wheat lines in Ethiopia because it would be very easy for example, for stem rest to blow across the border from Kenya, where it's already virulent to SR24. But there were a few lines that had that quantitative resistance. So it wasn't conferred by a single gene, but by multiple genes. And so those are designated with this APR here. And circled is kingbird, which was a variety released in Kenya previously that was highly resistant to the 4 races and doesn't have a single gene. It has the quantitative resistance. And so based off of these data, we were able to start rapidly increasing seed in Ethiopia. And there's a picture of a truck full of kingbird seed. And then it was released in Ethiopia in 2015 and started being grown there. So that appropriate phenotyping really helped us to make changes and to get resistance to multiple races. And the second part of what we're doing now and going forward is regarding using several genes. And so going back to our gene progress chart, so we have these 40 plus genes, how can we use several of them at the same time? Now what these circles represent are areas of the wheat genome where you have more than one gene at a very close position. And so one strategy that we've adopted is to create wheat lines that have 2 or 3 of these genes together at the same place, and then to backcross those multiple genes into advanced wheat breeding lines. So we call those linkage blocks. What that allows us to do is to get one place in the genome to integress into a wheat breeding line that carries multiple genes. And so in the United States, we've been delivering several of these linkage blocks to collaborating breeders, mostly in the northern United States, like in the Dakotas and Minnesota, where stem rest historically has been the most problematic. And so we're starting to see these genes come into the breeding pipeline, and obviously it's going to be some time before we see a variety with any of these genes, but that's what we've been working on. Now, the last research component I'd like to emphasize is regarding new strategies that are on the horizon. And for this, this part, I'd like to remind you that now we have several of these genes where we know the DNA sequence. These genes have been cloned. And so can we use different strategies and not just traditional crossing and back crossing and testing? And so cloning of these genes has become quite easy compared to how it used to be. And we've been using a rapid cloning or resistance gene isolation technique that it's outlined here. There are 3 main components to this pipeline. The first is that you take a wheat line that has a single resistance gene, and then you mutagenize it, and then find susceptible progeny that are derived from that wheat line that was resistant. And so basically you've mutagenized the resistant line and caused a change in the DNA sequence of the resistance gene. And now you have these susceptible lines where you know that the resistance gene has been mutated and there's been a change in the DNA sequence. Well, what we do is we extract DNA from these susceptible lines and then sequence the resistance genes that are in that line. And by doing that, we can identify a gene that is probably the causal gene. And this picture just shows here GABO56, which is a resistant line that Nirmala was working on in our lab. And she derived these 8 susceptible mutants from GABO56. And then when she sequenced these 8 lines, she found a single disease resistance gene, which is shown in blue here. And in that gene, in 7 of the 8 susceptible mutant lines, that gene had been mutated. So it's very likely that this is the gene that caused the resistance in that GABO56. So using this pipeline, we've been able to find a lot more genes that are conferring resistance. So finding genes is great, but how can we use these clone genes going forward? So traditionally, what I've been talking about previously is that we might have a cultivated variety. We do just a traditional intercross. We get an F1 between the 2 lines, and then we make all of these back crosses. So by the end, we get a cultivated variety that has a small piece of resistance part of the genome from this donor line. So this process takes a lot of time, but it's been what we've been doing. Now, if you have clone genes, you can start making genetically modified wheat that has the genes inserted into the genome. And so here is showing that the transgenic process where you take a vector where your gene is on a vector with a promoter, a terminator, sometimes a selection gene. And so these constructs will often have like 4 different pieces of genes from various different species. But you can take that, insert it into the wheat genome, and get a genetically modified variety that has a small piece of a foreign DNA that is now resistant. Also, you can kind of fine tune this process so that you're using sequences that are native only to wheats. You don't have all these different species of DNA on your construct, and then you insert the gene similarly into wheat and get an engineered variety that may or may not be technically genetically modified depending on the regulations. But then that can be used as resistance. So we know that if we just do transgenic or a cisgenic approach with one gene, that's really not going to get us very far, because we know the pathogen can become virulent to one gene. But since we've cloned so many genes, this opens up the possibility that we can make a construct and put 5 or 6 genes on a row on that construct, and then insert that into the wheat genome to get a line that has 5 resistance genes or so, and use that. So I think this is a very promising technique that you probably will see much more of in the not too distant future. However, it's still technically genetically modified, and I'd like to make the point that in the US, no wheat that is grown in farmers' field is genetically modified, and that's because we export a lot of our wheat to countries that do not want genetically modified crops. And so it's something that is nice, but we currently can't use it because of the regulatory environment. This is really unfortunate when you think of areas like Africa, where they don't have a lot of resistant varieties, and it would be really nice to be able to use these newer techniques to develop resistant wheat lines that should be durable. And so if you have the 5 genes together, this could be a scenario where the wheat line might not break down to the pathogen. And then finally, there's also genome editing, which we're starting to do here, and a lot of people are using as well. And there's really two different ways that you can use genome editing. You can basically make deletions at specific places in the genome. This is good if you want to, knock out a gene. This isn't really what we want to do with stem rust resistance. But what you can also do with genome editing is that you can make a very specific change in your genome to make your wheat line become resistant. And this process does not involve inserting, foreign DNA. It's a process that in the US is actually allowed. And so currently I don't know of any wheat grown in the field that is genome edited, but it's something that a lot of people are working on and you'll probably see more of in the future. And so using genome editing, we could go to different places in the genome and create, or make these genes resistant. So maybe we're not taking 5 genes in the same place and inserting them in, but you could do genome editing at several stem rust resistance genes throughout the genome to make a variety that has multiple resistance genes. So hopefully we can use some of these cloned resistance genes in the future for some of these new techniques, and this could really help us round the curve in defeating this pathogen once and for all. I'm optimistic that we can do that with these newer techniques. So we've talked about where we're going, our recent progress in phenotyping, the single race nurseries in Ethiopia, not just looking at UG99, but looking at other strains. And then I've talked about using linkage blocks and also the newer techniques. And I'd like to end with a few conclusions. And these conclusions are going back to those things that I wanted to emphasize at the beginning. So using technology and also international collaboration. And so if you go back to what Borlaug did in the Green Revolution, I mentioned that this Lerma Rojo 64 wheat varieties and other varieties that were used in the Green Revolution were disease resistant. And when you look specifically about what we now know about, these varieties, you can see here that this Green Revolution variety had 5 stem rust resistance genes that were effective to the stem rust pathogen at the time. And so there were 2 APR genes and 3 genes in red that were effective to the most virulent race at the time. So Borlaug, without molecular markers, without new techniques, was able to develop a wheat line, with 5 stem rust resistance genes. And so we're really still trying to catch up to what Borlaug achieved in the Green Revolution in addressing UG99. Borlaug was able to do this and to translate that technology into the field to make a big difference for people around the world. And now with what we've been working on, I'd like to emphasize the international collaboration. And this building is really unique. It's in Kenya. It's actually a stem rust inoculation building. I've never seen anything like this around the world. But this building, again, is from the 1960s, and it emphasizes the international research at the time between the US, Canada, and Kenya to invest in addressing stem rust. So this building has kind of fallen apart, but with the latest emphasis on UG99, there's been another big investment in different countries working together. Here's a water reservoir that's used for irrigating that nursery in Kenya. Here's a brand new greenhouse in Ethiopia that was built for doing greenhouse inoculations in the country of Ethiopia. We have the single race nurseries now up and running in Ethiopia. And this is a picture from the inside of our biocontainment lab in Minnesota, looking out into the winter. So we're able to have this facility to work with foreign isolates in Minnesota. So we have all of these great facilities, but it's important not to grow complacent. And I'd like to end with a quote from Borlaug, from what he wrote in that New York Times article in 2008. And he wrote, UG99 could reduce world wheat production by 60 million tons, but a global crop failure of this magnitude can be avoided. Before it is too late, America must rebuild, not destroy, the collaborative systems of international agricultural research that were so effective in starting the Green Revolution. And so with these words from Borlaug, emphasizing that we need to work together, there needs to be funding to address these issues that aren't just nationalist issues, they're international issues. We can make progress, and it's important to maintain our relationships, our work together in order to not grow complacent. And so hopefully 50 years from now, stem rust won't reemerge as another problem. With that, I'd like to highlight a lot of the people that are in my lab right now, listed here, Sam Stockson being a key technician, a lot of collaborators around the world, and here in Minnesota, funding from the USDA, USAID, and the Gates Foundation. And this is a picture of our high-tech equipment in Minnesota, harvesting some of our wheat plots there. And just leave you with an image of our team, and encourage you to work together and set aside differences and pursue the common goal. So thanks very much, Brad, and for all of you for being online here.
And thank you, Dr. Rouse, for sharing all that you did and the amazing work. When you hear the whole story, it's very impressive what you have done and what you continue to do in that international collaboration. We do have a few questions. We probably won't get to all of them, so I apologize if you did submit a question, but I do want to get to a few because we've had some really good questions come through. The first is from an individual who actually grew up in southeast Wyoming on a wheat farm where they produced hard red winter wheat. And their question is, what role has climate change played in the development of stem rust pathogens?
Well, I assume that, I was just thinking about whether you were referring to the wheat side or the pathogen side, but it sounds like you're really asking about the pathogen side. And I guess, fundamentally, we don't know for sure, but I can tell you that stem rust is a pathogen that is adapted to warmer temperatures. And so the years that we see epidemics are often the generally warmer years. And what's also kind of dangerous on the pathogen side is that we've seen that with UG99, not only is it adapted normally to warmer temperatures, but it seems to be adapted to relatively cooler temperatures compared to previous strains of the pathogen. And so, as the climate has changed, we've seen the pathogen adapt and become more virulent over time. When you think about projections for warmer wheat production in South Asia, that really worries me because historically stem rust hasn't been a problem in India, but if they get a relatively warm year, it could be quite devastating. And so we really don't know, and it's kind of scary. And so I think stem rust is going to be one of those diseases that we could definitely see more of in the future.
Thank you. Another question that we have is, besides plant breeding for resistance, are there any other techniques that can be used to control stem rust? Are there any agroecological approaches that can be used, perhaps in combination with resistant plant varieties that can control stem rust?
Yeah, great question. I didn't talk about fungicides, and that's currently a major method of control in East Africa. Problem there is they often can't get fungicides for various reasons. But with regards to agroecological things, there are a few things that you can do. So growing a variety that matures faster or planting earlier are potentially methods of control. This disease going back centuries was problematic, and what people have done is in Roman times, laborers would go out in the field with a rope and drag it through the field to kind of knock the dew off of the plants, and they thought that could actually help with the disease. And there, I didn't mention this at all, but you can eradicate the alternate host of the pathogen. And so stem rust also infects a bush called barberry. And there was a major eradication program, mostly in the 1920s in the US, to get rid of these bushes around wheat growing fields. So that's something that can be considered. The bushes are coming back in a lot of areas. But you know, the pathogen is very specialized. And so you've seen the pictures of the disease, but those brown colored pustules are full of spores. Those spores are airborne. They blow in the wind. They can blow even across oceans. And it makes it quite difficult to control once the disease gets going. So you can start with basically no visible disease and end up with devastation within a couple of weeks. And so, you know, I think you're a little bit limited with what you can do, but there are a few options.
Very good. And one question that came through, and actually we had, when it came through, I kind of had to smile a little bit because it was actually something we were discussing before we even started, but and thinking of some of the stories that you shared, but how has maybe COVID impacted your work, whether it be around the world or even in your labs right there at the University of Minnesota?
Yeah, it's impacted us quite a bit. Normally we are able to travel and evaluate some of these nurseries in Africa. And so we've had to rely solely on some of our collaborators there. And so that's been okay. A big problem is that we have restrictions on number of people who can access the lab. The nature of our research is very much doing work in the greenhouse and in the molecular lab. And so that's really cut our research progress back recently. So we're looking forward to whenever we can defeat the coronavirus and get back to full steam.
Absolutely. And once again, I just want to apologize for everybody. We're not going to get to all these questions. And I know we're getting close to the top of the hour here and so forth. So with that, Dr. Rouse, I just want to give you the opportunity, if you have any final words of wisdom or anything that you want to share and leave with us here today.
Yeah, you know, I would like to leave you with just a simple phrase that's kind of guided a lot of our work in our group, and it's from Borlaug. And he said, "Take it to the farmer." And so, you know, there's a lot of work that can be done, a lot of research, but what we really have, you know, wanted to do is have our research be applied in the field for people to be able to use it. And so, I don't know, you know, where you're at. A lot of you are, you know, various majors working on different things. And there's always kind of a attention to promote yourself, to maybe, you know, in our world, to just publish journal articles, or, you know, whatever it might be. There's always attention there. I encourage you to think about what's really important and to make an impact in our world.
Thank you for that. And we also want to thank the World Food Prize. We are so fortunate to host individuals like yourself throughout the years and learn from and engage with all of you. And so we appreciate that partnership here at Hawkeye Community College and the Global Agriculture Learning Center. It was also neat. I know a lot of our participants were from the US, but I saw some folks from Greece who had joined us. So we even had that international audience. We thank all of you for joining us today. And with that, I guess we'll take it to the farmer and go from there. Thanks everybody for joining us.